Hello BIOL A240 students. Today we're going to be discussing infection and epidemiology. We'll start out with a discussion about beneficial microbes, and then we'll move on to epidemiology, which is basically studying how those microbes are spread. We're going to start out with a quote by Robert Bauman, the person who wrote the book. Bear in mind that most microorganisms are neither harmful nor expressly beneficial to humans. They live their lives and we live ours. Relatively few microbes either directly benefit us or harm us. The idea that's being brought forth here is that microbes aren't specifically trying to harm us as hosts or trying to benefit us. They are merely trying to replicate. We've talked about metabolism, we've talked about replication, we've talked about how microbes gain nutrients from the environment to be able to do these things. If it happens to be that our cells hold the nutrients that these microbes need, then they will lyse our cells to get to those nutrients. If it happens that they can gain these nutrients without harm us, they will do so. But these microbes are not specifically doing this to harm us or to benefit us. Their only goal is to replicate. So that's where microbiota come in. Microbiota are the communities of microbes that live with or on a host and interact with that host. There are many different names for microbiota: normal microbiota, normal microbes, normal flora, beneficial microbes, indigenous microbiota, symbiotic microbes, microbiota. These are all basically the same thing, those communities of microbes that live on or in us. A little bit of vocabulary to start out. These are different types of symbiotic relationships as the book defines them. We have mutualism, in which both the first organism and the second organism, so here you can think of the microbe and its host, benefit. An example of this is bacteria in the human colon, where the microbes are able to gain nutrients and protection from the environment by living within our colon, and we're able to benefit from the nutrients that that microbe may provide for us, or protection from pathogens. Another type of symbiotic relationship is commensalism, in which one organism benefits and the second one neither benefits nor is harmed. Most microbes live in some sort of commensalism with other microbes. A great example is the mites on human hair follicles. They gain a benefit from nutrient sources and protection by living on our hair follicles. We are neither harmed nor benefit from this relationship. Amensalism is a relationship in which one organism is harmed and the other one neither benefits nor is harmed. An example of this is a fungus that secretes an antibiotic that kills another organism, but it doesn't gain anything from that second organism. And the last example is parasitism, in which one organism benefits from the relationship and the second organism is harmed. A great example is many of the potentially pathogenic microbes that we will be discussing for the rest of this class, including tuberculosis. This is a bacteria that's found in the human lung. It causes disease in the human lung, and it benefits from living there by gaining a great environment to survive. Some other vocabulary: resident microbiota. These are microbes that are with the host for the life of the host. Then there are transient microbiota. These can't persist in the body. Although they can't persist in the body, they may still change the microbe or the host, the other microbes that are there, or the host while they are part of the microbiota, but they don't actually live with the host or the microbial community for the remainder of the life of the host. And then there's the host. Who is the host? I keep referring to the host. The host is the animal or plant that the microbes live on or in. So we would be the host for the microbes that live in our body or on our body. And when you're thinking about the microbes that live on our body, think about the microbes that are on your skin, the microbes that are attached to your hair follicles, the microbes that live on the surface of your body. A great reference, a good source for more information on the microbiome of the humans, is the NIH Human Microbiome Project, in which several labs have worked together to characterize which microbes are present on or in humans, and what those microbes are doing. Now, there are many different types of microbes, and importantly, there are many different types of communities of microbes that live in different parts of the body. Now, I'm not going to have you memorize each of these particular types of microbes that are associated with each part of the body. And part of the reason for that is because not all the microbes are listed in this table. So for example, in the upper digestive tract, there are these particular microbes present in the upper digestive tract, but there are many more. There are hundreds of different species of microbes that are living in the upper and lower digestive tract. These are just a subsample of the different microbes that are present. Some of these microbes are found in multiple locations. So I'm not going to have you memorize those, but I do want you to consider the fact that you see some microbes listed here over and over again. A great example of that is Streptococcus. So here we see Streptococcus that makes as part of the microbiota of the human urinary and reproductive system. And here we see Streptococcus again in the male urinary and reproductive system. And we see Streptococcus here again in the upper respiratory tract. So these are microbes that are in several different places. Another great example is Staphylococcus, seen here with the eyes and skin. And we see it again, Staphylococcus here in the upper respiratory tract. We see it again here in the upper digestive tract. And so this is, and here it is once again with the female urinary and reproductive systems and in the male urinary and reproductive systems. So this is one microbe that's in many different places. Some of these microbes that are usually considered pathogens are part of the resident microbiota. We are going to discuss Streptococcus and Staphylococcus quite a bit when we talk about diseases of the human body. And yet here they are, they're part of the normal microbiota. Why is that? Well, one of the reasons is because we're listing genera here, and these have different strains within the genera, and some of those individual strains are able to cause disease within that genre, while others don't cause disease. So Staphylococcus epidermidis is a member of the skin microbiota, but Staphylococcus aureus can be a resident microbiota, but can also cause disease. Now, ignoring exactly which microbes are there, take a look at the notes for each of these different locations. In these notes, and you should read these notes, these discuss the different environments in which these microbes have to live. And these different environments indicate which microbes will be found in that particular area. So for example, the nose is cooler than the rest of the respiratory system and has, for that reason, unique microbiota. Whereas we see that the female urinary and reproductive system is a very acidic environment. And so the microbes that will be associated with that are able to live in an acidic environment. For example, the microbes of the skin live on the outer dead layers of the skin and follicles and pores of glands. And when we get to the chapter on the skin, we're going to learn that the skin is oily and salty and cooler than the rest of the body. So the microbes that will live there are going to be very different than the microbes, for example, in the upper or the lower digestive tract. I don't want you to memorize the different microbiota that are here, but I do want you to recognize that each microbial community is different from one location of the body to the other. Each body site is different. The microbiota includes viruses and bacteria and fungi. You can see here that there are many different types of bacteria that are present on several different surfaces, just as there are viruses such as the bacteriophages that are found in many different places as well. And then these fungi are different types of microbes that can live in different parts of the body as well. Okay, so where do you think these microbes come from? Just take a minute and jot down your thoughts. The microbiota come from many different places. They come from the vaginal canal when you're being born. They come from the doctors and nurses and other folks that are present at your birth. They come from the air, they come from milk, they come from formula, food, water, anything that's put in the mouth, really. They come from your clothes and your bed that you sleep on. These are considered fomites. They come from your fingers and your pets and dirt. They basically come from just about anywhere. And in fact, you can consider just about every source as being every surface is a potential source of microbes. And this is just a summary of the different places in the body that are colonized from different parts of the environment. So for example, the skin is often colonized from the vaginal birth canal as the baby is being born, and environmental surfaces, whereas the digestive tract is most likely going to gain its microbes from dietary sources or environmental sources. That doesn't mean that the gastrointestinal tract only gets microbes from dietary sources or environmental sources, but those are the two most common places that those microbes will come from. If you're interested, there's a great video on the microbiota from PhD Comics, and this is just a fun little video that talks about the microbiota and what we know about it and what we don't know. Now, these host-microbe relationships can be complicated. I like showing this example because this is an example in which a microbe is lives within a host and is beneficial for the host, but it can also be a pathogen for another microbe. So, for example, Xenorapidus or Photorapidus live within the intestine of the worm. That worm infects an insect, and then those bacteria are released and are able to kill the insect. So for the worm, it's a beneficial microbe, but for the host, for the insect, it's a potentially pathogenic microbe. And then the nematode is able to use the biomass of the insect as a source of nutrients to reproduce. So this is gaining a definite benefit from these particular microbes. And then these microbes recolonize in the new generation of worms, and then the cycle continues. So here's a great example where a microbe can be a pathogen, potentially pathogenic to one organism, the insect, but can be beneficial to another organism, the worm. So now let's focus on microbes that cause disease. Where do microbes that cause disease come from? One place is the microbiota. The microbiota, when it's where it's supposed to be, can be beneficial for the host or not cause any relationship either beneficial or harmful to the host. But if those microbes get into the place where they're not supposed to be, they can cause disease. So for example, here's microbiota that's supposed to be on the skin, but occasionally these microbes can get deep into the sweat gland, and then it can cause infections or pimples. Then there are normal gut microbiota. If normal gut microbiota is disrupted by let's say antibiotics, then you can get an overgrowth of one particular microbe like Clostridium difficile. So this is a microbiota that's part of the this is a microbe that's part of the normal gut microbiota, but when that microbiota is disrupted, this microbe can overgrow and cause disease. And then microbiota can also cause disease in patients for which the immune system has been suppressed. So for example, in a patient who has the advanced stages of the acquired immunodeficiency syndrome or AIDS, then members of the normal microbiota can cause disease. So for example, toxoplasma is a parasite that lives within cells of the host and typically does not cause disease as it remains in a stationary cyst form. But when the immune system is compromised, the parasite lyses out of that particular cell and then invades new cells, lyses out of those, invades new cells, lyses out of those, and so you get destruction of the tissue. But that only occurs when the immune system is compromised. Another way to get microbes where they're not supposed to be is the parental route, and this is an injection of normal microbiota deeper into the skin through needles. So here we're just showing 4 different types of needles, and we're showing that these needles can go through the skin and grab microbiota and then push these microbes deeper into the skin You don't need to know all of these different terms, just know that the parental route refers to the fact that these microbes are put deeper into the skin through needles. There are many different portals of entry for microbes. In addition to needles, there are microbes that can get in through the eye, the nose, the mouth, the placenta, the vagina, the urethra, the anus, the ear, the broken skin, or insect bites. So basically any way you can think of any opening or that's natural, or openings that can occur through other broken skin or insect bites can be a way for microbes to get in. This is showing that there are some pathogens that are also able to cross the placenta. So for example, if the mom is infected with toxoplasma while she's pregnant, then the parasite can cause toxoplasmosis in the adult, but it can also cause, it can enter into the embryo or the fetus and result in the death of the fetus, or microcephaly, or mental retardation, or other diseases in the placenta, in the fetus developing in the placenta. And there are a number of different microbes that can cross the placenta. These are just a handful of the different microbes that can cross the placenta to cause disease. Another potential source is animals. So animals can harbor microbes, and these microbes can either be beneficial for the animal or harmful to the animal, but when they are passed to humans, some of these microbes can cause disease. These are more difficult to eradicate because you have to be able to find all the animals that are infected with that particular microbe and either cure or vaccinate them so that they aren't able to spread that microbe to humans. Some examples are rabies, bubonic plague, and anthrax. And these can come through direct contact with the animal. For example, an animal that is infected with rabies can bite a person. That's direct contact. The rabies virus is then passed through the saliva into the infected host, into the human or other animal, and then it's transmitted to the human. It can be these microbes can be transmitted through waste. So for example, you can get toxoplasma from cleaning the cat box, which is why women who are pregnant are told not to clean the cat box so that they don't potentially become infected with toxoplasma. You can get animals you can get microbes from an animal reservoir by eating the animal, or from vectors. Vectors are insects or other organisms that are able to pass a microbe from one animal to another, or from one host to another. A great example is mosquitoes that can transmit a number of diseases from one host to another. So an example of a zoonotic disease is found in this article. This is a great article on zoonotic diseases. Specifically, this article discusses the spread of antibiotic-resistant microbes through animals receiving antibiotics in feed and healthcare in third world countries. So if you're interested in learning more about zoonotic microbes and the spread of antibiotic-resistant microbes through animals, this is a good article for that. These are examples of some different zoonotic diseases. So we are actually going to discuss many of these in the last 6 chapters. So if you want to get a head start on learning the names of these microbes, I would highly recommend that. And finally, we have 2 more places that you can get microbes, potentially pathogenic microbes from. One is human carriers. So for example, this is Typhoid Mary. She was a woman who was infected with Salmonella typhi. She did not have any symptoms of the disease, and so she didn't think that she carried this microbe, and she would prepare food for people who were susceptible to the disease. And when she would prepare that food, she would transmit that microbe from her to the new people. She did this with several families, and several families became sick when she prepared food for them. She infected 51 people. 3 of those people died. She eventually had to be quarantined and prevented from cooking for any other families. Currently, for Salmonella typhi, there are over 5,000 cases in the United States on an annual basis. There are 21 over 21 million cases in the world. This is transmitted by shedding in the stool. So in this particular case, typhoid Mary would use the bathroom, not wash her hands properly, and then fecal matter would be transferred from her hands to the food. Symptoms include high fever, weakness, stomach pains, headaches, and other symptoms. Other examples of human carriers, diseases that are spread through human carriers who are asymptomatic, so don't show any symptoms of the disease, are tuberculosis, HIV, and syphilis. But there are many others. And then finally, there are non-living reservoirs of diseases. So for example, there are microbes that can be found in the dirt that can cause disease in humans, water, biofilms that form on pipes. There are many non-living reservoirs of potentially pathogenic microbes that can cause disease. Okay, so those are normal microbiota and places that we get normal microbiota, and disease microbes that can potentially cause disease and where we get them from. So now let's focus in on those microbes that can potentially cause disease and the virulence factors that those microbes harbor. Virulence factors are something, a gene, a protein, an enzyme, that these particular microbes have that enable them to cause disease in another host. We have some vocab here again. This is a good list for you to think about, to learn, to study for the exam. So for example, these are prefixes or suffixes, and these are the meanings for those different prefixes and suffixes. So if I was to talk about somebody who had hepatitis, you would be able to go to the chart and you would see hepa means liver, and itis means inflammation of a structure. So hepatitis would be inflammation of the liver. Or you may talk about somebody who has idiopathogenic disease. The idio would come from unknown. The patho would come from abnormal. So idiopathogenic disease would be something where the cause is unknown and the causes disease in a particular organism. So what influences the virulence of a microbe? Many things do. So here we have different microbes that can potentially cause disease. Down here we have lactobacilli. We just saw that lactobacilli are part of the normal microbiota of several different areas of the body. So why would we say that it causes disease? Well, normally it doesn't cause much disease, and so that's why it's down here under less virulent. But as you go up here, up this chart, you can see that there are microbes that are more virulent. So for example, Francisella tulinaris is almost always pathogenic when it comes inside of a host, a mammalian host specifically. Yersinia pestis is another microbe that almost always causes disease when it infects a human. And in these particular cases, you don't need a lot of these microbes to cause disease, and that's what makes these more virulent. Many things can influence how virulent a pathogen is, a potentially pathogenic microbe is, including symptoms, including things that are associated with the host. So for example, the age of the host, the immune status of the host, the immune response to the microbe. Those are all things that are driven by the host and have nothing to do with the microbe itself. Others come from the microbe. So for example, the state of the microbiota. Is the microbiota a nice diverse microbiota, or is it dominated by one or two species? The genome of the microbe can influence whether or not a microbe is pathogenic. The environment can influence whether or not a microbe is pathogenic. So for example, some microbes can only produce toxins in the absence of air. And so if you have that microbe in a location that has oxygen, it won't produce that particular toxin. So the environment is really what drives whether or not that microbe will be pathogenic. And also the location of the microbe. So lactobacillus, again, if it's in the vagina or if it's in the digestive tract, it doesn't cause much disease. But if it gets in the body cavity where it's not supposed to be, it can have disease. There are a number of different virulence factors that are that come from the microbe itself. One is adhesins. A microbe needs to be able to adhere to a surface to be able to invade that surface. In this particular case, we're talking about a host. So microbes need to be able to adhere to cells to be able to attach to those cells, to be able to get into either into the cells or to cause disease. So a great example of that is the C. coli. The C. coli right here has receptors that bind to the mucous membrane of the intestine. And when the C. coli is able to bind to this mucous membrane, then it can cause disease. If the E. coli, for some reason, is prevented from getting to or attaching to the mucous membrane of the intestine, this particular strain of E. coli won't cause disease. And here we're seeing a close-up of a ligand on a microbe that attaches to a receptor on a host cell. This is the cytoplasmic membrane of a host cell, and this allows this microbe to adhere to the cell. Specificity is determined by the ligand receptor interaction. So this is a chemical reaction between this, either a chemical or a force reaction between this particular ligand and this receptor. And there's usually specificity that's associated with that. So this ligand most often will only associate with this particular receptor. Biofilms allow microbes to bind indirectly. So for example, if you have a biofilm that allows a microbe to survive within that biofilm, that biofilm isn't, the microbe isn't directly attached to the surface, but it's attached to the biofilm, which is attached to the surface. Other virulence factors include extracellular enzymes. Remember that enzymes are proteins that can be used to move a reaction forward, and they can be used over and over again. So extracellular enzymes are proteins that can be secreted from the cell. That's why they're extracellular. That can be secreted from the cell to perform a reaction over and over again. We have a couple of examples here. Hyaluronidase and collagenase are two enzymes that can be secreted from bacteria that are able to break through intestinal surfaces, break the bonds that hold epithelial cells together. So for example, here we have invasive bacteria that reach an epithelial surface. They release hyaluronidase or collagenase, and these enzymes break apart the proteins that hold these cells together. And now that these cells are not tightly bound together anymore, this bacteria can invade deeper tissues. Another example is coagulase and kinase. So here we have bacteria that it's producing coagulase. It's secreting the coagulase away from the bacteria. So it's an extracellular enzyme. That results in a clot forming. And the bacteria then later produce kinase that dissolves the clot and releases the bacteria. So both the coagulase and the kinase are required for this particular microbe to be able to cause disease. Then there are toxins. Toxins can be can also be secreted from the microbe. So here we have an exotoxin that's secreted away from the bacteria, like those extracellular enzymes. In this particular case, this exotoxin, it kills the host cell. So in this case, we have a cytotoxin, toxin referring to the fact that it's killing, and cyto referring to the fact that it's killing a cell. But you can also have endotoxin, and endotoxins are toxins that are associated with the bacteria. They are not secreted away from the bacteria. They're actually embedded in the membrane of the bacteria, so they're held tightly to the bacteria. Here we have LPS. LPS is the endotoxin that we're going to discuss in this class. LPS is usually associated with the membrane of the bacteria, but when the bacteria dies, that releases LPS, and now LPS has a part of the molecule that's normally not exposed that comes in contact with the immune system and results in inflammation, fever, diarrhea, shock, and blood coagulation. We're going to discuss LPS in great detail later. Here's a comparison of exotoxins and endotoxins. So exotoxins can come from gram-positive and gram-negative bacteria. Endotoxins come from gram-negative bacteria, not gram-positive bacteria. The relationship is for exotoxins is that they are metabolic products that are secreted from the living cell. Endotoxins, on the other hand, is LPS, which is a portion of the outer cell wall and the membrane that's released upon cell death. I'm not going to read through this entire table. You can take a look at this table, but these are things that you should know for the exam. Be able to talk about whether or not these different things are fever-inducing. So, for example, exotoxins are not fever-inducing, endotoxins are. But be able to discuss heat stability, effect on the host, antigenicity, toxoid formation or for immunization, and representative diseases along with chemical nature. Other virulence factors include anti-phagocytic factors. Now, we haven't talked about phagocytes yet, but phagocytic factors are things that enable a cell to engulf another cell. That's phagocytizing a cell. So you could have one cell that phagocytizes another cell. That means that the first cell engulfs the other cell, wraps around the other cell, takes it in, and is able to destroy that cell. Anti-phagocytic factors are factors that the bacteria or microbe can produce that prevents the cell from phagocytizing the microbe. So, for example, immune cells are able to phagocytize microbes and then destroy those microbes. But anti-phagocytic factors on bacteria or fungi or other microbes can prevent the phagocytes from engulfing that microbe and destroying it. Here we have a capsule that's bound to the bacteria, and we have a phagocyte. That capsule prevents the bacteria from being phagocytized by this phagocyte. You can have also incomplete phagocytosis. So in this particular case, we have a capsule around the bacteria, and the phagocyte has brought in that bacteria into a phagocytic vesicle. When this microbe is brought in by a phagocytic vesicle, the lysozyme needs to be able to bind to this phagocytic vesicle, and then this lysozyme contains vesicle contains lysozyme and other factors that are able to kill the microbe. But if the microbe is able to prevent this vesicle from attaching and binding to the phagocytic vesicle, then this lysozyme is not emptied into here and this microbe is not killed. So that's what we see here. In this incomplete phagocytosis, this vesicle has not bound to the phagocytic vesicle, and this bacteria is able to reproduce because it's not killed by the lysozyme. Okay, so let's think back to the first or second lecture in which we discussed Koch's postulates. Koch's postulates are basically a way to determine a set of steps that are used to determine whether or not a microbe can cause disease or is the cause of a disease. The first step is that the suspected causative agent must be found in every case of the disease and absent from healthy hosts. In this case, we're talking about microbes. So if there is a microbe that is not found in healthy hosts, but is found in every host that has a particular disease, then it's a suspected causative agent. The agent, or the microbe, must be isolated and grown outside of the host. That's in vitro. So the microbe must be able to be grown in vitro. When the agent is introduced to a healthy, susceptible host, the host must get the disease that the original host had. And then finally, the same agent must be found on the diseased or in the diseased experimental host. So you must be able to recover that microbe from that initial host. Now, I present these as though they're definitive, but they aren't. There are exceptions to Koch's postulates. So, for example, some microbes can't be cultured in the laboratory, so you would not be able to recover these and grow them in vitro. Some diseases are caused by a combination of microbes and other cofactors, including other pathogens. In that particular case, because in Koch's postulates you need to isolate one particular microbe, you would not be able to satisfy Koch's postulates with those microbes. Ethical considerations prevent applying Koch's postulates to microbes that require a human host to cause disease. We're going to discuss sexually transmitted disease. Many of those are specific only to humans. And so there are no animal studies to study those diseases in. And then finally, sometimes microbes can be ignored as potential causes of disease. Okay, so now we're going to get into epidemiology. I wanted to define for you two terms. One is etiology, which is the study of the cause of a disease, and the other is epidemiology, which is the study of how a disease spreads. So let's look at the stages of infectious diseases. Here we have a graph. On this axis we have time. So this is early time, later time. This can be days, or it can be hours, or it can be years. This time can vary from one pathogen to another. And here we have the number of microorganisms or the intensity of signs or symptoms of that disease. So for example, if this was a common cold, this would be the number of viruses that are causing the cold, or the intensity of the sneezing and the coughing and the clogged mucous membranes associated with that cold. For each disease, there's some sort of incubation period, and that's a time when there are no signs or symptoms of that particular infection. So at this point, we have a microbe that has infected a person. For a while, they're at a low level of low number of microbes in that particular host, and during that stage, there's an incubation period where you have no signs or symptoms. Then there's the prodromal stage. Prodromal period is a period in which the patient has vague or general symptoms. So for example, for a cold, that can just be feeling really tired. Those are really vague symptoms. The fatigue can come with lots of different diseases and may not be specific to that particular cold virus. Then there's the illness stage. At this point, the patient has the most severe signs or symptoms. So for our cold example, that's when your head is just completely congested, you're coughing, maybe sneezing a lot. This is when you actually have the signs and symptoms of that particular disease, and they're specific to that disease. And then there's at some point some decline in those signs and symptoms. And this is when, in that cold example, you no longer feel quite as congested, your head is starting to clear a little bit, maybe you're coughing less, you're not waking up in the middle of the night coughing. But in this particular case, you still have some signs of the disease. Maybe you're coughing a little bit during the day, but your signs and symptoms have declined. And then there's convalescence, and that's when you have no signs or symptoms. Now note here that you still have some of those microbes present. In that case, you're still able to spread that microbe to other potential hosts. So you would still be able to pass this microbe to other people. Now the incubation period for different microbes is different, so that's why we have a vague time here. So in the example of a cold, this entire timeline would maybe be a couple of weeks, but for some other microbes, any one particular stage can take a different period of time. So for example, in the incubation period, you can have some incubation periods that are really short. So for Staphylococcus foodborne infection, we're going to learn that a toxin produces this, and so the incubation period is really short. It's less than a day. But for other microbes, you can have a much longer incubation period. For the flu, it's about a day. Cholera, it's 2 to 3 days. Genital herpes, 5 days, etc., all the way down here to decades. So for example, for leprosy, the microbe that causes leprosy can lay dormant in the host for a very long time, from 10 to more than 30 years, and then finally cause disease in the host. Now we've talked about different ways that microbes can get into a host. All those same ways are ways for microbes to exit the host. Basically, any portal for humans can be a portal of exit. There are the eyes. You can cry and transfer microbes through the tears. Secretions from the nose, you're all familiar with the common cold, and mucus secretions, and those can contain virus particles that can be spread from one host to another. There's saliva or spit, there's milk glands and secretions in women, secretions from the vagina. Blood is another way to potentially pass pathogens from one host to another in humans. Urine, feces, semen, skin flakes, blood, earwax, even earwax can be a potential way to pass microbes from one person to another. Now, when you're passing microbes from one person to another or from one animal to another, what you're really talking about is transmission. There are many different modes of transmission. There's contact transmission, vehicle transmission, and vector transmission. Within each of these, there are different types of way to transmit diseases. So under contact transmission, this is really direct contact, like handshaking, kissing, etc., or indirect contact, like sharing a drinking glass. One person may use a drinking glass, microbes can get transferred to that glass, and then if somebody else takes a drink from that glass, the microbes can get passed into the new host. A great example of indirect contact that can spread disease is the common cold. And then there's droplet transmission, in which microbes are transmitted in the droplets from sneezing. This can typically is reduced to about 1 meter. That's about as far as water droplets can be transmitted. And there are many different diseases that can be spread through droplet transmission, like whooping cough or streptococcal pharyngitis, otherwise known as strep throat. Then there's vehicle transmission. Vehicle transmission requires something else to transmit the microbe from one host to another. And that can be airborne, like dust particles or droplets. And we saw droplets up here as well. Many different diseases can be airborne. There's waterborne diseases that can be passed in contaminated water, like Campylobacter or cholera or Giardia. And then foodborne diseases that can be transmitted from contaminated food, like food poisoning or hepatitis A, listeriosis, etc. Vector transmission requires something else to transmit the microbe from one person to another, and that something else can be mechanical, like the body of flies or the body of cockroaches. Some diseases that can be spread through mechanical transmission include E. coli, diarrhea caused by E. coli or salmonella, or trachoma. And then vector transmission can also be biological, like the mosquitoes that I talked about earlier that can transmit many different types of disease, or ticks or lice or mites. And these are some examples of of diseases that are transmitted by biological vectors. Okay, so now let's talk about the epidemiology of infectious disease. This is really going to discuss the transmission of or the spread of diseases. Again, there are some vocabulary words. We have acute disease, a disease in which symptoms develop rapidly and runs the course quickly. That could be something like the common cold that infects a person, and the common cold virus infects somebody and then is passed within a couple of weeks. There can be chronic disease. Those are diseases with usually mild symptoms that develop slowly and last for a long time. And we're going to see many examples of chronic diseases in the last six chapters. There are subacute diseases, asymptomatic diseases, latent diseases, communicable diseases that can be transmitted from one host to another, contagious disease, non-communicable disease, local infections, systematic infections. This is a good table for you to study for the exam. We'll be discussing several different vocabulary words. As I said before, a great way to learn the vocabulary words is using flashcards. So for example, you might want to write focal infection on one side of your flashcard, and on the other side, write infection sites that serve as a source of pathogens for infections at other sites of the body. Now, occurrences of diseases can be endemic, sporadic, epidemic, or pandemic. These all vary based on where these outbreaks occur and how frequently they occur. An endemic disease is a disease that's normally found in a particular area. So, for example, the Four Corners is endemic for plague. plague can commonly be found in the Four Corners here in the United States. It's a normal range. A new case of that disease is commonly found during certain times of the years. The official designation of this is a disease that's normally present in a particular region. Then there can be sporadic outbreaks. These are sometimes present in a region. They may happen annually. They may happen yearly. Sorry, it's the same thing as annually. They may happen every decade, but they don't normally occur. There's not an occurrence of that particular disease on a regular basis. Then there are epidemics. Epidemics are diseases that are present in one part of the world that's present in greater frequency than normal. So, for example, there may be outbreaks of salmonella or outbreaks of cryptosporidium in a particular region. It only occurs in that particular region, and it's an increase in the number of cases much greater than what would normally be found. But it's specific to one region or another, usually within one city or one country. That's different than pandemic. Pandemic is present in greater frequency than normal in several different countries. So in this particular example, we see a disease that's occurring both in South America and North America. It's not found all over the world. So for example, Canada and Alaska up here are don't have this particular disease, but it's found in multiple countries, and that's what makes it a pandemic. So here we have some more vocabulary: incidence versus prevalence. In the case of incidence, what we're looking at is the number of new cases that occur of any one particular disease. And in the case of prevalence, we're looking at the total number of cases that occur for any particular disease. The incidence may be the same as prevalence if a disease if a microbe causes a disease and then is quickly eliminated, but you could have new cases that result in an increase in the prevalence, but the number of new cases may go down. But because this is a chronic disease, the prevalence may remain high. An example of that is HIV infections, in which we had a large number of cases in the 80s and 90s that continued to increase, but patients were able to live longer and longer. And so now we don't have a large number of new cases, or for a long time we didn't have a large number of new cases of HIV, but because these people were chronically infected, the prevalence of that disease continued to increase. Now that's one type of chart. There are many types of charts that are used in epidemiological studies. These can include maps that show location of different outbreaks. So for example, here we have a map that shows, this is called a heat map, where you have different colors that indicate different numbers of cases in this particular case. So here we have cases per 100,000 population in the United States for this disease, random disease, and for Hawaii it's none. And you have different colors for all of these different concentration of cases, and in some cases you have more than one person per 100,000 population that's infected. Typically, the darker the color in these heat maps, the higher incidence or prevalence of a particular disease. We also have a time series here. So here we have a chart with time on the bottom and percentage of cases. So this tells us when a particular outbreak has occurred. So for example, there was an increase in the number of cases for this particular disease that increased significantly between July and September, but by November, the number of new cases has gone down. You can think of this as maybe like the flu, which occurs heavily in the fall, and then the numbers go down. And then you can have something that's more related to something about the patient population. So in this example, we have age group in years. In this particular case, we would have a disease that causes death in elderly people, but doesn't cause death in young people. Each of these graphs gives us some sort of information that's different for each of these diseases. And for any one disease, you would have many different graphs indicating location and time of year of outbreaks, or prevalence, or incidence, or age groups, or other factors. So for example, this could be the food that somebody consumed, the groups of food that people can consume, and whether or not that particular food correlated with disease. And in that case, you would be looking for the cause of a food outbreak. Okay, so I want you to take a look at this graph and think about what information you can get from this graph. So this graph shows number of cases of Legionnaire's disease. In light gray here, we have conventioneers. Those are people that attended a convention of the Legionnaire's folks. And down here we have non-conventioneers. These are people that didn't attend that particular conference. And on this axis we have time, so the days in July and August, and here we have the number of cases. So with that information, what can you get from this graph? So one of the things we can get from this graph is that there was a significant increase in the number of cases of people who had Legionnaire's disease in late July, early August. And it happened that the most frequent, these cases most frequently occurred in people who attended this particular convention, this Legionnaire's convention. Now, there were people who were not associated with that particular conference who had Legionnaire's disease. And from this particular graph, we can see that there's a possible outbreak. We can determine the date of that outbreak, and we can narrow down possible cause and location of this pathogen. So here we have the American Legion Convention. So what we can do is we can take a graph like this and see what particular things were occurring in that city around that time. In this case, there was an American Legion Convention in which elderly men were meeting together at a conference hall, a hotel, and this particular microbe that causes Legionnaire's disease was found in the water towers of that hotel. And so epidemiologists were able to determine that the microbe was being transmitted from the contaminated water in the water towers to these elderly men. And there were some cases from people who are not associated with the convention, and those were other people who were staying at that particular hotel or that came in contact with these, direct contact from these men who were also elderly. Okay, so here's a classic example of epidemiology. This is a map of London showing outbreaks of cholera. Any place where you see a red dot indicates a group of people who had cholera. The bigger the dot, the more people were involved in that particular cholera outbreak. Here in the middle, you can see this huge red dot, and it turns out that there was a water source that that people were using for their water. John Snow mapped all these outbreaks and saw that most of the outbreaks of cholera were occurring at this one particular water pump, and so he removed the handle from that water pump and then tracked the number of cases that occurred before that handle was removed and after that handle was removed. And removing the handle cut off that water supply from these people. And so when he removed the water handle, the microbe that was being transmitted in the water was no longer being transmitted to people because they weren't able to access that contaminated water. And so the number of cases went down. So does this case show incidence or prevalence of disease? In this particular case, we're talking about incidence because these are the number of new cases that occurred in this disease. Now, cholera is relatively short-lived, so this is actually incidence and prevalence. Now, hospitals can also be sources of outbreaks. In this case, I'm showing the spread of antibiotic-resistant infections in hospitals in 2014, a case study by the CDC. And the key points from this particular study showed that antibiotic-resistant bacteria can make infections impossible to treat because you're not able to treat with antibiotics. And these can be associated with hospitals. So HAI refers to hospital-acquired infections. In this case, 3 common HAIs were associated with catheters placed in a vein or a bladder, and procedures included these different procedures, and all of these happen to include catheters. And so this was a particular case in which this microbe, these infections, were being spread in a hospital. Now, one of the modern ways to determine whether how a microbe is spreading is to sequence the genome of the microbe. And so an example of that is the Zika virus outbreak that occurred in 2015. To determine where the infections originated and how they were spread, isolates of that particular virus were sequenced, and the sequences were compared, and it was determined that most of the people who were infected with this disease were travelers returning to Mexico from Colombia. And so they were able to determine that Colombia was the location of the outbreak. So what can we do with all this information that we get from these epidemiological studies? One of the things we can do is we can create new public policy. Public policy is basically determining what to do with when a particular outbreak occurs. One of the public policies is a reporting of the disease, and this is a hierarchy of where to report to. So for example, let's say that there are 4 hospitals that all happen to have an outbreak of a particular disease. Each of these hospitals report to a single county. In Alaska, we don't have counties, so each individual hospital reports to the state directly. But in the lower 48, hospitals report their diseases to counties, and then the counties report to the states, and then each state reports to the CDC. The CDC is a United States federal government agency, and so at the international level, CDC reports to the World Health Organization, or WHO. An example of the type of data that the CDC gathers can be seen in this MMWR, which is the morbidity and mortality weekly reports. And these are the number of reportable diseases. These are diseases that when hospitals see them, they have to report them to the CDC. The number of reportable cases, the number of cases of reportable diseases in each state. So for example, here we have Giardia and gonorrhea and Haemophilus influenza. And you can see from here in this MMR from 2013 that there were a total of 10,934 cases of Giardia in the United States in 2012. In 2013, there were 9,343. So there was a decrease in Giardia from 2012 to 2013. And you can see the number of cases for each individual state. From this, you can determine how many cases are happening in a particular region to determine whether that region is endemic for a disease, or whether new outbreaks are occurring in that particular region. And because this is reported on an annual basis, and you can determine whether new outbreaks are occurring and then make public policy based on these reports. Another example of public policy is quarantine. So for example, Typhoid Mary was quarantined. Quarantine just referring to the fact that she was not allowed to cook for other families. In this picture, we have Mary Mallon, who is Typhoid Mary, photographed with bacteriologist Emma Sherman. And this is at the site where Mary Mallon was quarantined. She had to live in the hospital. She wasn't allowed to go back home. And this is to stop the spread of this particular disease. And here you can see she was there for over 15 years after she'd been quarantined there permanently. Quarantines are used here in the United States sporadically. They're not used very often. Sometimes quarantines are used for infections like Mycobacterium tuberculosis. Mycobacterium tuberculosis is a communicable disease. So what does communicable mean? It means it can be transmitted from one person to another. And so here we have a transmission from one person who's infected with tuberculosis to another person. Because it's a communicable disease and is fairly easily transmitted, people are sometimes put into quarantine when they have tuberculosis because the treatment for tuberculosis is a lot of medication over a long period of time. So people are put into quarantine both to make sure that they're not spreading the disease while they actively have tuberculosis, but they're put in quarantine because they may spread the disease while they're on this medication that can take up to 6 to 9 months to cure the disease. And we'll learn more about that in a few lectures. Okay, so I have a discussion question for you. You're an epidemiologist working in Alaska. You've been keeping careful track of cases of antibiotic-resistant tuberculosis and realize that there's a sharp increase in the number of cases of tuberculosis that are resistant to all but one antibiotic currently available. The treatment requires 6 months of daily doses of pills. How do you contain this potential epidemic? I don't have an answer for you. This is something that's actually occurred in different states, and different states have handled this differently. In some cases, people are put in quarantine. In other cases, people are monitored closely and have to take their medication in the presence of a nurse at all times. In other states, nothing is done, and the person is told to take medication over the next 6 months, but not monitored while they do it. So different states can handle this differently. There's no one answer. There might be one answer that's better than others. It might be more effective to have somebody quarantined or to have somebody take medication in the presence of a nurse at all times. But there's no one way to handle this policy question. Okay, there are a number of really great resources to learn more about epidemiology. And I'm going to go through these. You don't have to sign up for any of these, but if you're really interested in healthcare systems and thinking about where outbreaks are occurring, monitoring outbreaks in your town or your city, or seeing where outbreaks are occurring in other parts of the world so that you can figure out if they're going to be coming to the United States or somewhere near you, in the near future. These are great resources. So one is ProMed Mail. ProMed Mail is a listserv in which you will get emails sent to you on a regular basis. I get 2 to 3 emails from ProMed every day that tells me about the different outbreaks that are occurring throughout the world. There's also the MMWR. That's the website that I referred to earlier. It's a collection of all the data that the CDC collects from hospitals around the United States. There are lots of news sources that produce really nice stories on diseases, including the New Yorker and New York Times, Science magazine. Not listed on here is a new newspaper or a new news source, particularly for health disease outbreaks, called STAT, that discusses not just outbreaks, but also discusses healthcare policies in the United States and internationally. That's STAT. And specifically, I have a couple of nice articles here talking about the microbiota. There's a lot of information out there on the microbiota. You can participate in research through the American Gut and the Kitty Biome projects. You can learn about the microbiota in this relatively easy to read and nice book called Follow Your Gut. You can also watch Rob Knight's TED Talk seen here on YouTube. And there are a number of really good books out there on the microbiota. And then I'm not kidding. These are 2 movies that are seriously good movies on epidemiology. And the band played on is a movie on HIV, how it was discovered, the public health policies that occurred around HIV when it was first discovered. Outbreak is a nice movie about an outbreak, obviously, of a particular disease and how a community handles the outbreak of that disease. Okay, that's it. That's all I'm talking about for epidemiology. I hope you enjoyed the lecture. Don't forget to answer the questions on Blackboard if you're one of my students, and I will talk to you soon.